Today we're going to be talking about mutation, and that's going to be changes in DNA that affect genetic information, that affects the way that um, a code is transmitted. Now, I've always been obsessed with mutations. You guys know that I used to breed snakes when I was in college, and some of these phenotypic mutations, they could make you a lot of money. Um, and it all depends on. Now, this double-headed snake isn't going to last very long, but it was a phenotypic mutation found in nature. You're never going to be able to keep this thing as a pet. It's just not going to last very long. But I was obsessed with the Teenage Mutant Ninja Turtles when I was younger. I was just obsessed with the fact, well, I was Donatello for 3 years in a row when I was 5, 6, and 7, I think. But I just love the fact that this little turtle that was supposed to live in an aquarium um, somehow grew up to be this ninja, you know, this ninja warrior that was able to beat up Shredder and his, I don't know, the bad ninjas, his Foot Clan, whatever that was called. But regardless, mutations are very interesting. There's a lot of developmental stuff happening right now. People are trying to figure out cures for cancer, and this is just a very big field, and it's only going to ramp up even more. Now, the two types of gene mutations we're going to talk about today are going to be point mutations and frame shift mutations. Now, a point mutation, there's going to be a change in one or a few nucleotides, and for this example, we're going to talk about substitution. Now, this is supposed to represent a strand of DNA, but the sentence over here is supposed to represent also a strand of DNA. Put it in threes so that you can understand that when it is transcribed, it does turn into mRNA, and this is how you read codons, but this is supposed to represent the DNA. Now, we're talking about point mutations in a strand of DNA. All right, so the original sentence reads, "The fat cat ate the rat." Okay, with this substitution point mutation, we can see that the only letter that has changed is H. Okay, so now that when we read the sentence, it says, "The fat hat ate the rat," it doesn't make sense. It doesn't convey the same message that we meant it. Now, in reality, there is no fat cat that ate the rat. I'm sure there is, but that's not what we're talking about. All right, we're talking about a strand of DNA where there's going to be a substitution point mutation. Okay, and the point mutation happens at this specific location. We can see that our original strand says CTG AGT. Now, after the point mutation, we can see that our strand is going to read CTG CGT again. And it's not necessarily always a big deal, but we'll talk about that in a little bit. The second type of mutation that we're going to talk about is going to be called a frame shift mutation, and the reading frame is going to shift. What does that mean? We'll talk about that also in a little bit. But there's two types of frame shift mutations. There's going to be insertion and deletion, and there is also chromosomal mutations. And what that means is, well, what it shows you is that there could be changes in an entire chromosome, not only just a segment, not only a gene, not only a certain part of DNA, but the entire chromosome as a whole. Those could be very detrimental to your health, or to someone's health that you care about. So, the first thing we got to talk about is the substitution is going to be missense. Okay, now substitutions will only affect a single codon. All right, but their effects could not be serious, but it could affect the amino acid, but that could be essential to the structure and the function of the finished protein molecule. For example, the quaternary structure of hemoglobin is that it has two alpha helices and two beta barrels. Now, that would be the quaternary structure. We talked about this when we talked about macromolecules back in October. But for this missense example, I just want you guys to look at the strand of DNA here. Okay, and this strand of DNA is it's going to start off like this. It's going to start off GGT, CTC, so on and so forth. Okay, the parts that we're looking at right now is going to be this letter and this letter. Okay, this letter and this letter. Now, with this substitution mutation, we could see that this thymine turned into an adenine, which is no good. Okay, not necessarily it's no good, but there was a change. Now, when we transcribe our original DNA, okay, and we transcribe it into our mRNA, we could see that our first codon is going to be CCA, CCA, proline, and our next amino acid is going to be GAG, GAG, glutamic acid. And guys, I'm just going to start saying the abbreviations. I'm not going to go into the entire name. We'll be here forever. Good. Sorry, just wanted to check that. All right, so back to my point. This is our original strand of DNA. Okay, this is our transcribed strand of mRNA, and this is going to be our protein. This happens in the nucleus. Okay, transcription happens in the nucleus, and then translation, it fits out of this nuclear pore, which you guys know from Bioman. It comes out and it finds a ribosome, and the ribosome is what makes our strand of amino acids, our aka our protein. Okay, but with this substitution mutation, we could see that this T has turned into an A, and when we go to translate this, we're going to get a different chain of amino acids. As you can see over here with the normal gene, we have pro, glu, glu, cis, gly. And over here, we're going to have pro, val, glu, cis, gly. Okay, so it did change our amino acid structure, our amino acid sequence, I mean. And necessarily it doesn't mean that it's going to be a very serious effect, but there could be an effect. All right, the next we're going to talk about, the next substitution mutation we're going to talk about is called a silent mutation, okay, aka synonymous mutation. But let's see how this would affect the amino acid chain. So we're going to use our same example again, okay, GGT, CTC, okay, it turns into proline, glu, cis, and gly. But with this silent mutation, okay, we did have a change in a nucleotide, okay? We did have a change in a nucleotide when we were transcribing our substitution. You can see that this C has turned into this T, okay, and also our codon has changed. Our codon is now GAA, and our original one used to be GAG. So when we do put this into our wheel, okay, GAG and GAA, GAG, glutamic acid, GAA, also glutamic acid. So this this synonymous point mutation did not do anything to the protein strand. This would have absolutely no effect on the phenotype. And where does this go away? Sorry. Yeah, so it's going to have no effect on the phenotype. It's going to have no effect on the protein chain. Both of these combinations, GAG and GAA, they code for the same amino acid. So if there was a change here, okay, it's not necessarily a very big deal. All right, but the code is different. The information, the original message is different. Okay, this is what a silent substitution point mutation is. Now, you're going to have something that's also called a nonsense mutation. And the way I like to remember this is that, and this one could be really bad, but if it's nonsense, it's not making any sense. All right, so as we could see, back to our normal gene, okay, we could read our codons, but now the codon that we're going to look at is going to be the second to last. We're not looking at that second one anymore. So, So GAA. All right. Now, we're looking at this code on here. Okay. And there was a point mutation at this exact location. Okay. And this is where this guanine turned into a T. A G turned into a T. Now, in the original DNA, a G turned into a T. So now our mRNA strand is going to also be different. But in this case, it's not a synonymous, it's not a missense, it's not coding for a different amino acid, it's not coding for the same amino acid, but in this case, it's telling the message to stop. Okay, so as you can see, pro, glue, cysteine, or whatever cis, and but we could see that we also have this glycine on the end. All right, now when we have this nonsense, this one that makes no sense, okay, it stops translation. So now we should have an amino acid that is 1, 2, 3, 4, 5. We should have a protein that is 5 amino acids long. But now we have a protein that's 3 amino acids long. So there is a problem here. In nonsense, those are serious. So now we have to talk about another substitution mutation. That's going to be called inversion. Okay, inversion mutations only affect a small part of the gene. All right, but it does affect the gene. Now, when we look at an inversion, what's happening here? Okay, there's no base that's being substituted. There's no base that's being changed. There's no base that's being deleted. But the thing that's happening is that these two nucleotides are switching places. So in this case, we have CTC, and in this case over here, we have CCT. So what did they do? They switched locations. They inverted their locations. Now, we can see that the rest of the mRNA strands are the same. CCA, GGA, GAG, UGC, GGU. All right. The only thing that's different is that these two bases in our mRNA are going to be changed as well. All right, and what does that do? Does it affect translation? Yes, it affects translation. Okay, so GAG, GAG, glutamic acid, GGA, GGA, glycine. All right, so yes, inversion can have an effect on a phenotype of an organism, okay, because necessarily making that protein is what is important, and our amino acid chain has changed. So now we're going to get into frame shift mutations, and that's going to be either and that's going to be when an extra nucleotide is either going to be inserted or deleted. All right, and what that does is it causes the entire genetic code to shift. All right, so using our sentence again, we're going to represent insertion. Okay, the fat cat ate the rat. Okay, but instead of this A, we added in an X. And what does that do to our code? Now our code says the fat cat zat lot doesn't make any sense. Okay, era T, not period T, but it is era T. Now, so this could be a very big problem. Okay, and also on the other hand, instead of an insertion, there could be something. There could be a deletion. Now I have to get this to go away. All right, but in a deletion, we could see that the fat cat ate the rat, and the fat cat ate the rat. Or it says the fat cat ate their at. Okay, now there was a nucleotide that was deleted, and it was right here. So let's look at these pictures real quick. Now we could see that at this location right here, there was an insertion of a T. Okay, there was no, it didn't change. It's not it's not the right nucleotide, but it was inserted at this portion of the code. Okay, so now that when we go to transcribe our mRNA and translate it into proteins, okay, there is going to be a problem with our reading frame. It's going to make no sense now. And these are very serious mutations. All right, same thing with the deletion. You can see that this A is in between these two G's, but if we take that A out of the equation, okay, we're going to be left with 5 nucleotides instead of 6. Therefore, we'll only have 1 amino acid that is translated at the end by the ribosome. Now, insertions. All right. An addition of a nucleotide could cause a frame shift mutation. And this is going to be another example. So as we can see, we're using the same normal gene throughout this entire presentation. The one that we're going to be talking about here is going to be the CTC portion of this normal gene. All right, and our mRNA portion is going to be GAG. So in a perfect world, this is what it would look like. All right, but with this insertion frame shift mutation, a base is going to be added. Okay, and where's the base going to be added? It's going to be added right here. Okay, it should say CTC, but instead of CTC, now it says GCT. And what happened is that we changed our reading frame. Okay, so in this case, when we put it into our wheel, GAG, GAG, I don't know why I changed anything, it's glutamic acid. All right, GAG is going to be glutamic acid, but when we come over here, okay, and this guanine was inserted, it changed our entire reading frame. You can see that we also have one hanging off in the end, and like I said before, you remember your translation assignment, you can't have 5.66 codons. You can't have 5 2/3 codons. You either have 5 codons or you have 6 codons. Right? That is how the ribosome reads the mRNA. That's how the anticodon is produced, and how the anticodon grabs the correct amino acid and brings it to the ribosome, and that's how the ribosome puts together the protein. But okay, as we could see, adding this G here, okay, the G was added right here. They added the G here, and what that did is it changed our entire amino acid chain. Okay, now we have pro, arg, gly, val, and arg. Okay, it's not even like they're very close. They're very, very different. All right, and that happened all because we added a nucleotide into one portion of our code. Again, right here. Now, deletion. I'm sure that you guys could put 2 and 2 together and figure this out, but at this point right here, okay, first we're looking at CTC. Okay, so it should be CTC, and instead that this thymine was deleted, okay, and it was taken out of the picture. But now our reading frame, okay, our set of 3 nucleotides, that reading frame has changed. We've taken out 1 nucleotide, and look, we're 1 nucleotide short. Okay, and again, you have to have 3 nucleotides to make 1 codon. So the ribosome doesn't know this. So if you take 1 out, it's just going to read the next nucleotide. So in this case, the nucleotide, the codon that it's reading is now GGG. Right? What's GGG? GGG, glycine. All right, so GGG is glycine, and what happens? Glutamic acid now changed to glycine. Next should be another glue, but no, we have serine and alanine. All right, so again, okay, this deletion of a nucleotide, this specific nucleotide here, this deletion, what it did is it caused a frame shift, and that frame shift did change our amino acid strand, our protein. Changed it quite drastically, actually. Not only did it change its sequence, but it also changed the number of amino acids on the chain. All right, so now we're going to talk about chromosomal mutations. All right, and when you guys are thinking about this, I want you to think about crossing over. Okay, you guys had great videos on on crossing over. Some of them were so professional it was scary, and but most of you guys did a really good job. Now, chromosomal mutations, there's going to be a change in the number or the structure of the entire chromosome, not just a segment of a gene, not one nucleotide, not even one codon for that matter, right? But the entire chromosome. Now, you're gonna have to follow me here. All right, so this is going to be our original chromosome, all right, and it's going to be ABC, and then next to it is going to be DEF. Makes sense, it's linear. Okay, with deletion, we're losing our B. Okay, we lost our B with our deletion. Okay, now these are not letters, these are entire chromosomes or pieces of chromosomes. Then there's duplication, ABBC, DEF. Okay, we can see that there was a duplication of this B portion of the chromosome. Now there is something called inversion, and it follows the same principles of what we just went over. Okay, but what happened was that this BC switched places with this DE, so they're actually backwards in the wrong place. But I can't really explain this to you, not being in class with you. If you take this little segment and you literally turn it around, yes, you will not be able to read the letters the same way, but they will be in the right place. So it's kind of around this pivot point right here that they just kind of invert each other. And the worst one, not the worst one, the most confusing one is going to be translocation. Okay, in translocation, things are going to be in different locations. Okay, so we should have ABC and then DEF, but now we have another section of the chromosome in our portion. All right, so deletion is going to be a part of the chromosome is going to be deleted. Duplication is going to be a part of the chromosome is duplicated, and inversion, the chromosome is going to twist and it's going to invert the code. That's what I was trying to show you. I got to think about a good way to show you that. Excuse me. And translocation. It's going to be genetic information is going to be traded between non-homologous chromosomes. Now, we've talked about homologous chromosomes, but we didn't talk about non-homologous chromosomes. From your meiosis videos, you guys, again, I just said it before, you guys did excellent on them. But when you had that tetrad forming and those two homologous chromosomes are coming together, they're homologous because they have the same gene sequence and about the same location, blah, blah, blah. But there also could be something called non-homologous chromosomes, and that's where it's just a chromosome next to another one. It doesn't necessarily have to be for the same gene or the same attribute that this organism is trying to express. So this is just some pictures in case you're a visual learner. With a deletion, boom, you can see that B was deleted. With a duplication, you could see that B was duplicated. Made our chromosome a little longer. Now, inversion. Okay, inversion, you could see that the B and the E switch spots. But translocation, this one's the hard one. So this is going to be that non-homologous chromosome. This is the one that we just talked about. Now, this is going to be just a little segment of that non-homologous chromosome. And that is going to put itself inside of this section right here. And now you can see that we went from a chromosome that was 6 sections long to one that is 8 sections long now. All right, what causes mutations? There's going to be 2 ways in which DNA can become mutated. Okay, it can be inherited, okay, from the parent to the child, or it can be acquired. Now, there can be environmental agents, mutagens, pesticides, tobacco, UV radiation, nuclear radiation. Now, the pesticides, just a fun fact, if you look at any of the fruit that you eat, there is always a sticker on the fruit. And if you see that first letter on letter, you hear me? If you see that first number is a 4, that means that they use pesticides to keep that fruit or vegetable. I don't think there's stickers on vegetables, but they keep that fruit, growing properly and not, infested by bugs or any other kind of animals trying to eat them. But if it has a 9 on the sticker, that means that it was truly organic and it was grown with no pesticides. That was just a little fun fact I learned the other day from TikTok. So tobacco can cause mutagens. It could cause a mutation, UV radiation, and also nuclear radiation. And there also just could be a mistake when DNA is copied. So it can be inherited, it can be acquired. But the way that it is acquired is it could be from a mistake when the DNA is copied or an environmental agent, but an inherited mutation, that's coming directly from your mother, your father. Now, the significance of mutations. Now, most of them are going to be neutral, okay, like eye color or birthmarks. Not a big deal, okay? Some could be harmful, like sickle cell anemia. And sickle cell anemia is caused by a point mutation. Some could be beneficial, right? So people that are sickle cell carriers are, they don't have to worry about getting malaria, contracting malaria. I'm not telling you to go roll around in the jungle, but they're resistant to malaria. Now you can see, looking over here, that this is, and you guys know I love to draw this on the board, but this is an RBC, an erythrocyte, and it doesn't have a nucleus. And the reason it doesn't have a nucleus is because that hemoglobin molecule we were talking about before kind of sits right here with its little active group on top. It's an iron active group, and that's how it attaches to the oxygen and brings it around your body. But we could see with sickle cell anemia that the red blood cells are just a little bit different shape. Okay, they kind of look like crescent moons, and they kind of get stuck in these little arterioles and vanuoles. And yeah, that's a big problem, guys. All right. But mutations are not all bad. They do give us genetic variation, and they give us crops, polyploidy. Now, the other day we were talking about meiosis, and I wrote on the board, and I was trying to drive a point home. I wrote 2N, and then the first division of meiosis, then I wrote N, and then that second division I wrote N again. And somebody came around and they wrote 3N, but which would mean that an organism had 3 copies of genes, or 3 copies of its DNA. But guys, this is a real thing. 3N. A strawberry is 8N, I think. And that's just about the number of copies that it has in its DNA. I can only imagine the bananas. I'm gonna have to look that up, and I'll have to get back to you guys about that. But yeah, that's for different types of crops that we have. Sickle cell anemia. All right, it's going to be caused in a change of one of the polypeptides found in hemoglobin. Now these are just better pictures. These are kind of old, from 1997, but they look nice, and I love the word erythrocytes. I don't know why. But this would be a regular erythrocyte, and these would be sickle cell erythrocyte. Now, sickle cell disease. It's going to be caused by an abnormal hemoglobin molecule. Like I said before, there is 2 alpha helices and 2 beta barrels. Take your pick, which one you want it to be. But with sickle cell anemia, it's going to form long and flexible chains. So this hemoglobin is not going to do what it's supposed to do. Now, this iron is supposed to sit right where my red pointer is. All right, and this iron atom is going to attach to the oxygen. It's going to travel all around your blood. But with sickle cell anemia, the hemoglobin makes chains, okay? And you're not going to be able to pull around 1, 2, 3, 4, 5, 6 oxygens around. All right, but just to give you some of the background on sickle cell disease, about 1 in 5 African Americans have it. 1 in 500. I'm sorry. It slows down blood flow and clogs small blood vessels. Its symptoms include anemia, weakness, poor circulation, impaired mental function, pain, and a high fever, sometimes even organ damage. Advantage: resistance to malaria. It kind of feels like we're watching one of those commercials for one of those drugs that are terrible for you, from one of the pharmacies with all disclaimers at the end. Yeah. So I'm gonna put this. This is a good place to end because I'm gonna put this on canvas. You guys are gonna be able to see it. Please let me know what you think about the screencast. I want to do this more often. It looks like we're going to be out of school for a little bit longer. And guys, please remember, all right, think for yourselves. Don't be sheep. I miss you guys. Hopefully I'll see you soon. All right, bye.